Great. So welcome to the Research Innovation Spotlight on Oncology. I'm Steve Gillison with the Alliance for SoCal Innovation. We put these on to connect folks from industry with the really great leading researchers in the region. We do this every few months. We pick a very specific topic. We get an amazing moderator to help pick the presentations from the universities in Southern California. And it was very difficult this time to make the selection. So I appreciate Steve Curtis, our moderator today, and helping that, as well as his associate Farnaz, who was great in looking through also to see who was an American Cancer Society grantee. We have several of those with us today, so we're really appreciative of the collaboration. And speaking of, and here's Farnaz right now entering the room. So, and speaking of collaboration, we do this in collaboration with Bioscience. They're a great regional partner of ours for all things bioscience. And we have here Shri Bachu. Hey, Shri, how's it going? You want to tell us a little bit about what Bioscience LA is up to?
Absolutely. Thank you for the introduction, Steve. And it was a pleasure. Thank you to the entire Alliance team for putting this together. Our friends at Alexandria and our friends at AACS. I think it's Bright Edge, if I'm not mistaken. I apologize. Dave and Jessica couldn't be here today. So as third choice, I'm very happy to be here. It's really happy. To give you guys a little bit of background on Bioscience LA, our main mission is to ensure Los Angeles has a collaborative, well-coursed ecosystem. And the overarching goal for Bioscience LA is mainly, if you're an entrepreneur, you're a startup founder, how can you get connected to the resources that LA has to offer? LA is super diverse, super vast, and it's very difficult to navigate that pathway. And Bioscience LA kind of wants to be that one-stop shop. You come here and you get connected to everyone. So if anyone would love to learn more, I would love to chat, and I'm your guy moving forward. It's a pleasure to meet everyone.
Great. Thanks, Shri. Yeah, great partners. We've had some events at their space, which is a wonderful resource for the region to have a meeting place and a hub for all things bioscience in LA. So if you're from LA or if you're passing through, you gotta stop by. With that, what I'd like to do is briefly have everyone introduce themselves. I will call on you, and you basically just say who you are, who you represent today. And we'll do that to make sure everyone understands who's in the room, because we also love the connection. Love for you all to connect with each other. We'd love to connect with the researchers. Everybody, this is a nice community here. We make sure we take advantage of this opportunity to connect. So what I'm going to do is I'm going to call on you, and then I'm going to also say who's up on deck, so you're not surprised when I'm just not cold calling. So and then when we're done with that, I'll have Steve Curtis take it away from there and all that. But for right now, I think we have Patricia Beckman, who would like to say hi, and then on deck is Aaron Denny.
Hi, I'm Patricia Beckman. I work with a company called BioStrategy, doing work with early stage companies, and also with the Cancer Fund. I'm in Seattle.
Oh, great. Welcome.
And hi, Steve.
And you know all about rain, then. You're like, you Angelinos. Yeah. Okay, Aaron, you are up, and on deck is Anthony.
Hi, everyone. My name is Aaron Denny. I lead Oncology Search and Evaluation at Amgen within our Business Development Group. I'm really excited to be here today.
Thanks, Aaron. Anthony, you are up, and on deck is Pingwei Feng.
Hi, Anthony Bajoris, founder and managing director of the Cancer Fund in Phoenix. So no rain here, usually.
That's great. Pingwei, you are up, and on deck, Alice.
Oh, I'm Pinghui Feng. I'm from University of Southern California. I'm a herpes virologist working on metabolic enzymes that regulate the immune response. Thank you.
Great, thanks for joining. Alice, you're up, and then Lauren, you are on deck.
Hi, everybody. Alice Pomponio, Vice President for Innovation and Impact Investing at American Cancer Society. I don't have my logo because I didn't want to steal Steve's thunder, and Managing Director of Bright Edge, based in Boston.
Great, thanks. Lauren, you are up, and on deck is Carlo.
Hi, everyone. I'm Lauren Bradley, the Associate Director of Corporate Engagement at the Jackson Laboratory. We are a biomedical research institute, and oncology is one of our focus areas for basic discovery.
Right, thanks, Lauren. Carlo, you are up and on deck. Oh, Farnaz, we can hear from you. Hi.
Yeah. Hi, everyone. Nice to meet you here all. So I'm Carlo. I'm the Director of Chemical Biology and CTO of Armida Labs. Armida Labs is a University of California, Riverside spin-off company founded by Professor Maurizio Pollecchia, and we are focused on the target delivery of chemotherapy, lysocovalent agents, and fighting cancer resistance mainly. So we are kind of a new company, probably 2 years old or less. Exciting to be here.
Thanks, Carlo. Fernaz, you are up and on deck as Catherine. Oh, Fernaz, I can't hear you. I think maybe something's on mute. Should we come back to you? Yeah, still can't hear. But all right, we'll come back to you, Fernaz. Sorry. Catherine, you're a moderator emeritus. To see you again. And
Great to see you, Steve.
Yeah,
this is awesome.
Yeah, and Carl, you're on deck, by the way. So.
Hi, everybody. I'm Catherine Zavala. I'm the COO of MedTech Innovator and also the Managing Director of BioTools Innovator. BioTools is our new program dedicated to life science tools and diagnostics. Really great to be here and to see some old friends, Aaron and Patricia. Nice to see you guys. Thanks, everyone.
Great, thanks.
Can you hear me now?
Oh, yes, we can. Hi, Fernaz.
Oh, excellent. Thank you. Sorry about that. Fernaz Bakshi, Director of Innovation at BrightEdge, the Impact Venture arm of the American Cancer Society. I work with Alice and Steve.
Great, thanks. And thanks for all your help. I gave you a shout out earlier, but thanks for your help with curating the speakers today. Thank you.
Yeah, no, my pleasure. Thanks for having us.
Yeah, great. All right, so now Carl, you're up, and CT, you're on deck.
Hi, great to meet everyone. Carl Hess, Managing Director at Outcome Capital, a highly specialized healthcare and life sciences advisory firm and investment banking firm here in Boston.
Great, welcome. Hi, CT, you are up and on deck as Sisio.
Yeah. Hi, everyone. My name is Chian Tran. I'm based in the Monterey Bay area, but I'm with Resilience, which is a high-tech CDMO based in 12 different locations in the US and Canada. And my role is Senior Director of Strategy for Cell Therapy. Nice to meet you all.
Great, thank you. And Sisio, now you're up, and Ryan, you are on deck.
Yes, good morning, everyone. My name is Sisio. I'm from Amgen. I'm a Global Development Lead in Development Research.
Great, thank you. Ryan, you're up. I love the new glasses by Ryan, and on deck is Divya.
Thank you, Steve. It's good to be here and see everyone again. So I'm Ryan Witt. I'm an investor with a group called Tech Coast Angels, and also I'm helping a company called Renovo RX, which is a cancer drug device combination in a phase 3 right now and publicly listed. So that's my background. Love to invest in deathly things like cancer. So hopefully not so deathly for long. Thanks to everyone here.
Thanks, Ryan. Divya, you are up and on deck as Daniela.
Good morning, everyone. I am Daniela Castanocco, the Director of Strategy, Research and Development in Duarte, California, a city of hope. And I am happy to be here and looking forward to the presentation, to hear the presentation.
Great, thank you. All right, so Divya, now you're up, and Seth.
Hi, I hope you guys can hear me now. Hi, my name is Divya Sadar. I am from Loxo Oncology at Lilly. I'm a project team lead, and I'm here in the New York site for Loxo. It's nice to be here, and thank you for this invite. I actually have another colleague with me who would also like to introduce himself. He did register a little bit last minute, so I hope this is okay.
Yeah, absolutely, sure thing.
Hey, this is Carsten. I'm director for immunopharmacology team and our organoid team. And as Divya mentioned, we are based in New York City, and we're focusing on our oncology discovery efforts. Nice meeting.
Great, great, thank you. Great. 2 for 1 on one screen. That's perfect. That's very efficient. Thank you. And Seth, I guess, not last but definitely not least, how are you?
Hi, everyone. I'm Seth Lieblich. I'm a principal at HVC, where we do early stage seed, Series A, Series B investing.
Great, thank you much. All right, now I will hand it over officially to Steve Kurz for the remainder, and just to, I guess, remind everyone the format, just so Steve doesn't have to go through that stuff, I'll hand it over to Steve in a second, is that each presenter will speak for about 10 minutes. And then we will then go into questions and answers. I would love for you all to ask questions. If you do not have any, Steve has many up his sleeve, but this is meant to be a great conversation. Would love to hear, from everybody, and fellow presenters. You can ask questions of each other as well. This is just it's good have a good conversation. And with that, I will leave it to Steve.
Great. Thank you, Steve. So as mentioned, I'm Steve Curtis, Director of New Ventures at BrightEdge. BrightEdge is the impact venture capital arm of the American Cancer Society. Maybe just to give a brief overview of what that is and what that means. So as I'm sure many of you know, the American Cancer Society has been supporting research, advocacy, and patient care in cancer for over 100 years. Many of the things that we are doing are focused on health equity and to make sure that treatments, screening, et cetera, and patient services delivery is accessible to all. It's kind of why the you can see my finger. The new tagline for the American Cancer Society's logo is "Every Cancer, Every Life." At BrightEdge, what we are looking to do is deliver mission through our impact investing efforts. We think we can find ways to invest in great companies that are squarely on mission for the American Cancer Society and drive both impact and financial returns for the society that way. We have a portfolio of about 16 or 17 companies. Now we've been doing this for a few years, but really undergoing a bit of a relaunch and expansion sort of at the moment. Myself and Fernaz are part of a team expansion just starting this year, along with a couple other colleagues. As we are growing, a big push beyond what we're doing on the sort of traditional investing side, if you will, is to create and expand our innovation services. We have a network of grantees, a couple of which are on this call and presenting today. And we would love to be able to support those grantees that have commercial aspirations for their technologies that are moving across that valley of death, as it were, right, of traditional grant funding to translational research and into the company creations area. So watch that space. Lots of exciting things to come in the future. But that's a little bit of who we are, what we're doing, and really appreciate the opportunity to participate in this call. Based out of Boston, since we're talking about the weather, it was in the mid to high 70s here yesterday. Almost felt like I was in Southern California. For Boston in November, that is crazy. Got a strong dose of reality. It's in the 40s today. So back to where it should be up in the East Coast. So I'm jealous of all of you joining us from sunny California.
Agree, Steve. Yesterday it felt like we were in Palo Alto or someplace else suitably.
Right. Yes, certainly not Boston in November with temperatures in the mid 70s. All right, well, without further ado, definitely want to get right into the agenda. Thank you again to the Alliance for SoCal Innovation and Bioscience LA for organizing and putting this on, and of course the sponsors that make events like this happen. So kicking us off today is going to be Brian Eliceiri, Professor of Surgery at UC San Diego. And Brian's going to be discussing his genetic technologies that he's using to analyze immune cells and inflammatory reactions. Brian.
All right, well, thank you for having me. I'm going to share my screen here in just a moment.
Oh, actually, Brian, before you get started, one other little bit of housekeeping. As Steve mentioned, definitely want this to be interactive. Each presenter is going to present for about 10 minutes, and we'll have about the same amount of time for Q&A. If you'd like, feel free to drop those questions in the chat as you think of them. I can, call folks out or relay those questions to the presenters when we get to the Q&A section of each. Certainly feel free to come off mute and ask those questions, but if you're more comfortable, feel free to drop them in the chat. I'll be monitoring that too.
All right. Well, excellent. Well, first, thank you all for the invitation. It's wonderful to have this opportunity. And what I want to do is give you a quick rundown on an interesting project. Start with genetics and ends with a pharmacological target. And so this is my contact info, and I am at UC San Diego. And let me get the advanced thing going, right? Okay, so what is our target? I'm going to jump right into it. I'm going to give you some background on the molecule of interest, some of the immunology and some of the genetics that we've used to date, just to give you a sense that we've got a pretty full, complete approach on this strategy, but really focus on the potential for translation with a small molecule. So what is our target of interest? It's the alpha-7 nicotinic acetylcholine receptor. Shorthand is alpha-7 with the NACHR. or by its gene name ChRNA7. What you really need to know and the takeaway here is that there's a good crystal structure. It's 5 subunits. Different colors are shown here. It's a ligand-gated ion channel. It's very important in neuropharmacology, and there are knockout mice for it, and they're small molecule agonists. Why does that matter? It's a well-defined molecule. It has a well-defined biology, especially in the neurosciences, and there's knockout mice to test loss of function and small molecule agonists to explore the potential of activating this for protective effects. What are they? If you want to get a little bit more detail, the alpha-7 forms homomeric pentamers, and it, along with alpha-9, has some interesting structure. There are various different kinds of nicotinic receptors that I won't talk about today, and the general canonical function of these is to function as a neurotransmitter at neurojunctions. But what has come of late the last few years, and work primarily from the Kevin Tracy's group on Long Island, is this idea that alpha-7 is expressed on immune cells, in particular on macrophages in sepsis. And through a series of interesting stories that I won't get into any particular detail, this idea that T cells can be a source of acetylcholine that can act upon receptor expressed on macrophages. What does this do? It starts to change the thinking of a neurotransmitter as something functioning just in the neuronal signaling space, neurons, to something that may be important and relevant in immune cells. And there's a whole series of papers that really build this idea that the alpha-7 nicotinic acetylcholine receptor, ChRNA7, is important in signaling, mediating NF-kappa B signaling, TNF-alpha. And this led us to think about what its potential might be in cancer. And so just to jump right into it, what did we do? We set up some tumors in mice that are lacking the alpha-7 receptor. And these are tumors that are syngeneic. It's all a C57 compatible model. These are E0771 tumors, for those of you who are curious. And what we can show is that the loss of the alpha-7 gene leads to tumor permissive. And this is consistent with the general idea that stimulation of the alpha-7 in the sepsis paradigms may be protective, but in this case, decreased survival if you're missing the receptor, and increased tumor burden if you're missing the receptor, larger tumors, that sort of thing. So this is important genetic data. Why look at alpha-7? Well, this is something that has been characterized and actually quite nicely through the ImGen data set. So you don't even need to rely on us. And there's actually quite a bit of value in being able to demonstrate that expression of your favorite molecule would be in the kind of cell type that really changes the way you think of these paradigms, and namely expressed in bone marrow, spleen, and in particular in dendritic cells, and also in monocytes. And so we explored what the, how do we measure this in tumor-associated immune cells? This is something that has not been done to date. So we use things like, there are all kinds of interesting reagents like a Bunger toxin, which actually comes from a snake venom that binds the alpha-7 receptor, and we can show that it's present in spleen as a control and at a preferential level in CD11B positive monocytes, macrophages, as opposed to T cells. You can see it in the blood and not the knockout. We use a knockout as a control. And that's how we kind of gate the specificity of these reagents and can show that it's most highly expressed in the highest level CD11C positive cells in, say, a tumor, as opposed to the low cells. So these are various strategies to essentially demonstrate that this is a molecule expressed in myeloid cells, and in particular in tumor-associated CD11C high cells, which points to this idea it may be more relevant in antigen-presenting cells of one sort or another. Here we can show that the expression of MHC2 is reduced on the knockout cells, reduced CD11C positive cells, reduced infiltration of CD8 positive cytotoxic T cells. So these are all kind of the metrics that folks look for when it comes to tumor biology, and to make the case that there is a role for this molecule in a nice, elegant loss of function model. It's loss of function in the host. Here we can stain for C11C and show a reduction in tumor margins, MA, compared to the knockout, further suggesting that there is a role. Now, what I haven't told you is that the alpha-7 is thought to activate the JAK-STAT signaling pathway with transcriptional readouts being relevant. So we isolated the CD45 positive cells and can show changes in various chemokines in the infiltrating CD45 positive infiltrating cells between knockout and wild type. Some things go up, some things go down. So we said, well, can we model this in vitro and collect and culture bone marrow derived dendritic cells and do some single cell RNA sequencing and see if we can really map some clear differences in the program that this gene may be controlling. Because remember, this has not been defined previously in the cancer paradigm. And actually, this takes the understanding of alpha-7 signaling to the next level in the sense that these haven't actually been worked up even in the sepsis paradigm. So now here we jump to, that's really all introduction. Okay, so now we're going to talk about this agonist. And the agonist that we have focused on is the AR-R17779. This is a drug developed by AstraZeneca. It's off patent. It's been around for a number of years. This is a paper from JMed Chemistry in the early 2000s that basically does an elegant job of demonstrating the specificity of this molecule for the alpha-7 nicotinic receptor, as opposed to other heterodimers that are important and present in neuronal junctions. And this is because our sense from AstraZeneca's research interest was development in neurobehavioral disorders. And then this is essentially a mapping study for those of you who are thinking, tell me more about the organic chemistry. I'm not an organic chemist, but I know a bit. And so this is to demonstrate there has been some thought given to it. And the reason we picked this drug is because this is the one that has the most transparent clarity and depth of the organic chemistry for us to have some confidence of the specificity and in terms of the mapping of the structure itself. So what did we do? We now set up another, well, we use the same tumor model, the E0771 model that I mentioned before, the same one to match the knockout studies, and then explore, is there any increase in survival with the administration of the AR drug? Again, this is testing the idea that if the loss of this gene is permissive for the tumor, then if we can add an agonist, a small molecule, maybe it's protective and increase survival. And the answer is yes, we can. Are these huge differences? Well, not the most impressive in the world, but they're there. And there's a decrease in wet weight. And we say, well, okay, there are a lot of things that are out there in the literature that do this sort of thing. So we turn to another model, the 4T1 tumor model. It's a model of triple negative breast cancer. It's known to be very aggressive. It's now in the BALB-C strain. So now we're essentially saying, well, that's great in the C57 paradigm, but let's take it to a model that might be more commonly used. And what you can see is that the tumor volume is suppressed in the AR model, excuse me, the 4T1 model, the treatment with the AR drug, it's shorthand, and that the lung macrometastases is reduced. So we said, that's great too. So let's go for another model. Now, this is a genetic using the spontaneous onset in the MMTV promoter driving the polyomidal T. And these tumors get multifocal breast cancer. And what you can see is that there's an increase, my apologies, an increase in survival with treatment with the AR drug. And so then we thought, well, let's do a little pilot study and see if we were to add an immune checkpoint inhibitor to go along with the AR drug, playing on this idea, if the characterization of the immune cells suggests that there's some role for the alpha-7 in antigen presentation, dendritic cells, that sort of thing, maybe there would be some cooperative effect. And in this small pilot study, the answer was yes. So then we set up a full-blown study, basically using the 4T1 model, and approaching the question is, does combination therapy increase survival? So these would be the controls. This would be the anti-PD-L1, which is not known to be particularly effective in the 4T1 model. Here's the AR drug. They were all had to be euthanized just because they were alive, but the tumors were rather large. They had not died yet, though. And if we added the combination, we saw really a quite striking increase in survival. Now, this is the kind of data that really got us excited. And we thought, now this is important and really worthwhile sharing with an audience like this. And I hope you agree. And so what's the model? The model is, well, we're just hacking off from the kind of classic models for T cell activation, and then add in this idea that the alpha-7 pathway may be a way to modulate a kind of antigen presenting cell. I mentioned the JAK-STAT pathway. It may be affecting PD-L1 expression for example. And what are some things that we get asked? What are other alpha-7 agonists? This is not first-in-class drug. And these are the drugs recently reviewed, mostly in the area of nicotine addiction and behavioral. And many of these are affecting the either agonist binding site or the allosteric binding site. And this is the structure of our drug that we are using. As I said, it's off patent from AstraZeneca. This is a follow-on drug from AstraZeneca, which presumably is something that they're going to follow up on their studies. We don't know. We haven't been in communication with them. We are focusing on our publication, and of course, we have a patent disclosure. This is a compound from Bristol Myers Squibb. Again, for the organic chemists out there, these quinnucleotides. This is their kind of equivalencies in the structure here. And so a lot of similarity, very small changes. So naturally, we're thinking about maybe we could modify it. Here's just a quick search of what's going on. It's a very slow progress in clinical trials, mostly in Parkinson's. And so again, not really much going on in terms of cancer. What are we doing about it? Well, we've received pilot funding from the California Breast Cancer Research Program. We have an NCI grant pending. We have intellectual property pending around the method. And what we're looking for is translation, some kind of industry partnership, maybe some VC funding, where we could have some IP around the composition. And we have some ideas on what that might look like. What we often hear is, well, if it's not first in class, then call me later. Here's my contact information, and here's our team. And again, thank you all for your time.
Brian, thank you for the presentation. Definitely some really exciting science. Before I start with questions, let's turn it over to the audience and see if we can make this interactive. And I want to jump in with the first question. All right, I'll go ahead and get us started and get us warmed up. Brian, can you talk about agonizing the alpha-7 receptor? What potential toxicities might we be concerned about with this mechanism?
Right, yeah, that's a good question. So we have done some dose escalations in other studies unrelated to cancer, and we can go 5 times higher and don't seem to have any deleterious effect. We don't know about toxicity studies in humans, but presumably AstraZeneca does know. And these have been published for behavior studies in rats, and that's in older literature. So we are optimistic that the toxicity is low, and if we quintuple it, we don't see any adverse effects.
And the dosing that you are utilizing in your oncology studies is similar to those that has been used in the neuro setting. Is that correct?
Yeah, we've gone on the lower end. It's a 1 mg per kg dosing.
Okay, it's even lower. Yep.
And yeah, no, but those are good questions. And naturally, we should, once we have a paper in press, reach out. And the idea was to communicate with an audience like this to give some guidance. We've talked to some folks. Some will say, run with that as is, call me when you get the paper in press. Others is explore some drug screen and find your own version. But we already have a drug, so there should be something we can do with that. And therefore, we're following kind of the classic NCI funding path.
Yep. Denise, oh, I saw you raised your hand, Denise. It looks like you dropped the question here in the chat. You want to
chat here.
Come off mute?
Sure. I was just wondering if it's agonizing the nicotinic receptor, if it might be addictive like nicotine.
Well, I don't know to answer the question. Yeah, I don't. One thing I'll say about nicotine, for those of you familiar with the alpha-7 receptor, even though it's called nicotine because of muscarinic and nicotinic and how these things were originally described, nicotine is not a major agonist for the alpha-7 receptor. So we don't really equate nicotine and and the AR drug with respect to this receptor. So it's interesting, but maybe not necessarily that relevant for us. It may be addictive for other reasons, but not that we're aware of.
All right, Brian, I got one last one there for you then. Okay. Oh, was someone coming off mute to ask a question?
Yeah, my question was, could you maybe talk through the possible resistance mechanisms here that you think might arise?
resistance to the cells that survive after the therapy?
Yeah, resistance to the agonist specifically.
Mhm. Yeah, so that's an interesting question. We haven't really set up, say, a resistance study. Our thought is that our data indicates that we're affecting the host. And so if there was escape from the immunosurveillance, then those kind of classic standard type selection pressures would be there. But we're not thinking that there's any kind of direct effect of this agonist on the tumor cells themselves. So, but we don't know. We haven't really explored. We haven't set those kinds of assays up.
Great. Brian, maybe just one last one. Do you ever see genetic abnormalities of the alpha-7 in cancer? Is that something that is mutated since the knockdown is more permissive?
No, that's a good question. There isn't a literature of nicotine receptor mutations in smoking, but that's in tumor cells. And I'm not really, the simple answer is that I don't know.
Okay.
We're not intending to be like the be all end all for nicotine and cancer. That's a literature that's really old. It's out there and we don't proclaim to be experts on that. It's this idea that there may be immune cell targets that may have been overlooked, and there's a small molecule that we can rip off from essentially the behavior world and apply it to a model, and we need help on the organic chemistry or the IP. That's the ask. If anyone has ideas, let me know. Otherwise, we're going to go the same old route. It's going to take, time and persistence, and we've got some of that.
Hey, Brian, very quick question. This is Daniela. There are activators of these receptors. Have you ever checked if you activate the receptors, if you increase metastasis?
Increase of the alpha-7 receptor?
Yes.
Yeah, so the idea is that when we treat mice with these agonists, we have decreased metastasis.
No, I'm talking about treating them with an activator. There are activators of the for the expression of the alpha-7 receptors. So would you be able to see the opposite effect?
Yeah, so you said the word expression. This is an agonist for the receptor. I don't know that it increases the expression of the receptor. We haven't looked into that. Do I answer your question? I feel like I'm not. So
you could positively modulate the alpha-7 receptors, right?
Yeah, that's what we're arguing we're doing with an agonist.
Okay, and if you would do the opposite, you would block it?
Right, so that's an interesting question. So there are inhibitors, and we have not tested that to see if we could show pharmacologically what we're seeing with the knockout.
Could you do it with cells in culture and then just do a FACS plot and look for changes in expression?
Yeah. Now, we have not done a lot with the drug treatment agonist in vitro or with an antagonist, and these are all good questions.
Okay.
Interestingly, my peer review that has not come up. So just to give you an idea, mostly I get
Brian, I don't mean to cut you off, but I do, as moderator, need to make sure we stay on time and have time for everybody else.
Questions. Thank you all. I appreciate it.
Thanks much, everybody. All right. Thank you, Brian, for that and for all the great questions. Glad we're getting engaged and energized here. So next up, Crystal Marconett, Associate Professor of Translational Genomics at the Keck School of Medicine at USC, is going to be talking to us about long non-coding RNAs as potential surrogate biomarkers for tumor mutational burden. Crystal, take it away.
Thank you. I assume you can hear me. Is that accurate? Yes. Okay, great. I thank you, everyone, and thank you for inviting me to present this morning. Like the wonderful introduction I just got, my name is Crystal. Most people just say, hey, you, and start talking to me. So, feel free. Obviously, I'm running my own lab here, and we received a grant from the ACS to be able to pursue research in lung cancer. We're specifically looking at the interaction between total mutational burden and the immune system and how we can potentially predict better patient application for existing therapies. So my lab uses a combination of bioinformatic and molecular biology techniques to inform research questions. I have training in both, right? So we're really after this idea that, and it's a really hot topic right now in, treatment of non-small cell lung cancer. It's the largest histological subtype of lung cancer, largest cancer killer out there, right? Where we have this really amazing therapies, right? But they work by blocking inhibition, right? So it's negative. And this is a two-step process, right? In order for the immune system to recognize these tumors, not only do we need to remove that inhibition, but the tumor actually needs to be recognized as a tumor, right? So that's going to involve neoantigen presentation on the surface by MHC class 2, potentially presentation. So we have this situation where we want to have hot tumors where the immune system can recognize them, but not every patient has that. So we want to translate these really great findings. Immunotherapy has doubled the median survival time in lung cancer, as shown on the right. But only 20 to 40% of patients are actually going to respond, right? And of those who do have indication for PD-L1 therapy, they're positive for PD-L1, not all of those are going to respond positively to that therapy, right? So we're looking at that other arm. This is a study out of the Netherlands looking at various waves of immunotherapy in those who are failing and dropping off even after indication for it. So there exists a need for additional criterion, right? Who is actually going to benefit? We've got these two areas. So we're really looking at biomarkers of disease response that can be utilized in combination with the positive PD-L1 staining to improve the application of immunotherapy to patients. The area that we're going after initially was, well, in order for a tumor to recognize that, or the immune system to recognize the tumor, right, it needs to have something that is not normal in the cell, right? So these high mutation, we're expressing them off of tumors that have lots of mutations are going to produce peptides that have sequences that the immune system was not trained on, right? So the immune system could potentially recognize high tumor mutation burden, right? But that requires the actual tumor cell to take those mutant peptides and present them on the cell surface. So it needs a functional MHC class 2 antigen presentation system to be able to do that, right? Okay, so the first thing we did was we went after, well, can we find very cancer specific cell types? Sorry, the sun, it comes and it goes. Very cancer specific gene expression patterns that could be used as a surrogate biomarker for total mutational burden. It costs a lot of money. It's computationally intensive. Outside of academic medicine, it's not particularly well applied to look at total mutational burden, right? There's all of the sequencing involved. You have to realign everything to the genome. You have to call mutations. You need to have pipelines for that. And it costs money, right? And it costs time and it's an effort, right? That's not standard of care outside of big medicine in local clinics, right? So we're looking at something that can be easily and quantifiably measured. So we discovered this gene, and on the advice of counsel on this call, I was told to label it as link X. I apologize. Where we saw that on the advice of counsel, I defer all questions. Where we saw this particular gene was highly associated with mutational burden. When it is lost in the tumor, we get a lot more mutations. So we've started looking at that in a functional relationship for it, but what does that say about application for immunotherapy? So we looked at this gene in a lot more detail. And again, we need functional MHC class 2 presentation in order to put those neoantigens on the surface so the immune system can recognize it. So what we saw was that there was a positive correlation, i.e. when link X is expressed, a lot of these MHC class 2 genes are also being expressed, which would allow for these things to be put out there. We saw this in the big data sets that are publicly available that people can look at. We saw that in vitro when we would reintroduce this link that we're studying and then look at the expression of these guys. Just the mere presence of this link was able to turn on MHC class II expression in our cells. So that was really exciting. Another thing that is known to turn on MHC class II expression is interferon gamma treatment. So we treated ourselves with interferon gamma that actually turns on link. So they are functionally connected. And this is a critical component for that second arm of, positive response to immunotherapy. Looking broader, like, well, what is it a unique function of this gene? Is this a characteristic that we see across the board? It is not, actually. It seems to be a pretty particularly linked to this specific gene. What we did was we looked across all of the genes, the entire transcriptome, and correlated the expression of those genes in these large data sets to mutational burden. So things that are positively correlated to mutational burden are all DNA damage repair. Your cells have lots of mutations being accumulated. They're trying to repair that. All these things get turned on. There's this big ramp up, right? Conversely, things that are turned off when there's high TMB are almost universally immune system stuff. And the link we identified is one of the top genes across the transcriptome that's associated with that phenotype, shown here. So which immune populations is this associated with, right? Which immune cells are we actually going after? So we looked at the association of different immune signatures to see if there was patterns that emerged from the data. That's shown here. We're looking at different types of T cells, either inhibitory or stimulatory, to see when our link was expressed or when it was absent, was there a difference in composition of immune populations within that tumor? And what we saw in general was that inhibitory states, which would be reflective of a cold tumor, right, were enriched when link was silenced. We also looked at whether or not this was correlated to PD-L1 expression, right? That's that major indicator right now that's clinically applicable. About, depending on which staining panel you're using and what marker you're actually, like what assay you're actually using to look at PD-L1, it varies, but about 40 to 50% of patients will stain positive for PD-L1. So we wanted to see what the relationship between that gene and our link of interest was, and there was a significant relationship between them as well. So, okay, so our model is this, and this is going to be a little shorter. So our model is this, is that when our link is expressed, you have MHC class II presentation, and you're able to present neoantigens on the surface. And then the immune system is able to recognize that situation and attack. Whereas when the link is silenced, you no longer have MHC class II presentation. So even though we saw an accumulation of mutations and potentially a higher mutational burden in those, right? We saw a higher mutational burden. So we would think we're making more neoantigens. If you can't get it to the cell surface, it's not going to be able to be recognized. And the immune cells aren't going to have that thing to go after, right? So that's really where our model is right now. We've got, we're following up with that on the, the basic biology side of things. But the big take home here is that we are looking for a partnership with industry where there is access to a retrospective cohort of patients where we have RNA-seq level expression data. Keras won't do it because they don't have, it's not RNA-seq bulk. It's looking at exomal regions, which are protein coding genes. This gene is not in that list. Where we also have from those patients immunotherapy history. So we can retrospectively determine whether or not the expression of this gene was able to predict patient response. So that's a real ask right now. We've talked to a couple of companies. This data set is apparently very hard to come by. We're trying to generate it in-house right now. But as one can imagine, trying to de novo create an entire patient cohort at one institution of the size required to be able to do a study like this is difficult. So that's why I'm here today, to see if we can, collaborate with somebody who already has something like this going, and maybe get an answer to this really cool question. So with that, I would like to acknowledge the American Cancer Society and also the Department of Defense Lung Cancer Program for providing funding for us to pursue this. Thank you.
Great. Crystal, thank you for that presentation. I'll go ahead and kick it off again with questions, but I hope the audience jumps in as well. When you looked at expression of your link here of interest and PD-L1 expression, you show the correlative, but I'm wondering if there's a sort of additive value, right? Where you see high PD-L1 levels and link expression, right? Does that give you sort of a better potential predictor of response then to PD-1?
Fabulous question. We have not done that. We have not done that yet, but that's great. Well, yes, we can do that with the data that we have presently. So that's on the list of things to do. Right now, we're looking for that subset cohort where they have high PD-L1 expression, and they have, it's either positive or negative link expression, right? So we would want to see then the ones that fail and the ones that succeed. And we would predict based on our research that the expression of link would be able to stratify those two populations.
Yeah. Being able to have the outcomes data on PD-1 treatment and how that would be even
That's
more,
really what we're after.
That's really what you need.
Big data sets, the TCGA and other large data sets that are out there, we're all in the early 10s, right? And this therapy came online around mid-15. So it was there's only a couple of samples here, a couple of samples there. That's just really not the power that one needs to be able to answer this question. So we're looking at, trying to partner with somebody. This is an outstanding question in the field. Someone must have this data. Hopefully we can share. You know what I mean? Yeah. Okay.
Anybody else want to jump in? I have one more if not. Crystal, can you just maybe give a sense of what turning this, the assay that you're using in your lab to something that could be used as a diagnostic sort of in the more commercial setting? How easy or difficult is it to look for a link? Is that a staining of slides? Is that an RT-PCR?
It's a PCR assay. It's a PCR assay that, is easy to implement clinically. Yeah, thank you.
Great. All right, thank you, Crystal. Next up, Anna Wu, Professor and Chair of Immunology at City of Hope, is going to talk to us about engineering antibodies to deliver radioisotopes for both therapeutic and diagnostic applications. Thank you, Anna.
Thank you. Let me just put this into presentation mode. Can everyone see the slide?
Looks good.
Okay. Yeah, so thank you to the Alliance and to Steve and Steve for inviting me to participate in this forum and to tell you a little bit about our work on engineered antibodies for targeted delivery of radioisotopes. I'm actually going to focus mostly on radiotherapy, although a lot of our work leading up to this has been in the imaging applications. And I'm actually going to give you an overview and the take-home message first. And so what we're interested in doing is taking antibodies that recognize a target called prostate stem cell antigen and using them, adapting them as delivery vehicles for radioisotopes and in particular for therapeutic radioisotopes. So this is basically a way to do targeted radiation therapy in your patient. So it will be able to be delivered systemically and even traffic to those micrometastases that may not be visible, say, on your conventional imaging. So the hypothesis, of course, is that we can make engineered anti-PSCA antibodies for delivering therapeutic radionuclides. And this is a target that's upregulated in prostate cancer, pancreatic cancer, and bladder cancer, all of which are major unmet needs. And this is the agent over here on the right that I'm going to describe in greater detail in the upcoming slides. But just to give you a snapshot of where we are right now, we actually have all the key assays in place in terms of characterization of the protein, establishment that it's appropriately immunoreactive. And a little bit of history, we started with a mouse monoclonal antibody, but over the years, in collaboration actually with Rob Reiter at UCLA, we humanized the antibodies, and then we generated a series of engineered antibody fragments that are more suitable for delivering radioisotopes. And at this point with this particular lead candidate, what we can show you is that we want to use it for therapeutics, and we can demonstrate targeting and imaging in xenograft and knock-in models. And also I'm able to show you some preliminary data of therapeutic efficacy of this in a xenograft model in mice. So all in all, we think it's a very promising candidate in a field that needs new therapeutic modalities. And I think our major challenges might be further optimization of pharmacokinetics and dose delivery, because of course we need to take this into humans and see how it performs first. And then of course, prostate cancer is a very competitive field now. There are a lot of new therapies that have been recently approved and also under investigation, but this is the state of the field right now. And we have a patent application filed on this agent. So first I want to step back and take a look at the bigger picture and why we're looking at prostate and pancreatic cancers as our target diseases. If you have early stage prostate cancer, those patients do very well. The patients who have advanced disease definitely need alternative therapies. Once the disease has spread, there's only a 5-year survival of about 30%. And recently there actually have been a slew of developments in therapies for metastatic prostate cancer. And it includes targeted radiopharmaceuticals, such as the new PSMA 617 small molecule that's radio-labeled with lutetium-177. It's known as Pluvicto, and that was actually just approved in March of this year for patients with metastatic disease. And it's very promising in terms of inhibiting or eliminating tumors initially, but disease will still recur. And in addition, one of the challenges with using that particular agent is that it also localizes to the salivary glands and the lacrimal glands. So there are side effects, and patients can get xerostomia, which means loss of saliva, which can really impact their lives. So we do need some alternate treatments. And then in pancreatic cancer, the need is even greater. It really is one of the most lethal cancers. You can see that, we get 57,000 new diagnoses each year, and then 47,000 deaths every year. And at this point, the 5-year survival is really only around 9%. And one of the reasons for that lethality is that the disease is typically advanced at the time of diagnosis, either locally advanced or already has spread metastatically. So there is a huge unmet need here for novel systemic therapies, and targeted radiopharmaceuticals, again, could be a potential modality that can be effective in this disease. So let me tell you a little bit more about the target we're working on. It's called prostate stem cell antigen. It's a cell surface glycoprotein that shows low expression in normal tissues and organs, including the prostate, the bladder, and the lumen of the stomach. But it's highly overexpressed in prostate cancer, almost all prostate cancers, and a significant majority of pancreatic cancers as well, as you can see in these immunohistochemical stainings here on the right in prostate and pancreatic cancers. And so in that sense, it's very complementary to the existing PSMA-directed therapies. And this is a target that's been known for a while, and there have been attempts to use it employ it as a target in cancer therapy. And for example, over the years, anti-PSCA antibodies have been evaluated in pancreatic cancer. There's a PSCA CAR T cell program. There are programs both in the US and in China, including one at the City of Hope. And then others are developing bispecific antibodies that target PSCA and, for example, recruit T cells to tumor. And there are a number of those types of engineered antibodies in clinical evaluation. And finally, there are a number of groups that have explored vaccination against prostate stem cell antigen as a potential cancer therapeutic. So it's not a completely new target, but yet there's still a lot that could be done with this target. So here's our technology concept. My lab specializes in engineered antibodies, and in particular, engineered antibody fragments that we've optimized for delivery of radionuclides. And the reason this is important is that we start with intact antibodies, and they can have wonderful high specificity. They can show excellent tumor targeting and localization and uptake in tissues of interest. We know, from the slew of approved antibody therapeutics that they do hit their targets and they can be efficacious in patients. But in general, they have slow blood clearance, which is good if you have a biologically active antibody. But if we want to think about specifically delivering a radioisotope, a therapeutic radioisotope, that long half-life becomes a negative because bone marrow in particular is very sensitive to radiation, and that sensitivity limits how much activity you can administer to a patient because that radioactivity is in the circulation for weeks and it's constantly exposing the bone marrow. So that's a limitation of the intact antibody. And so what we've done is we've kept those binding sites, bivalent binding to the targets of interest, but we re-engineer the rest of the antibody to suit our applications. And for a while we focus, I'm going to skip down to this bottom one, which has sort of been our prototype mini-body antibody fragment, which has 2 single chain Fv binding sites from a parental antibody, but only has a CH3 domain and disulfides so that it's a covalent molecule. And so this is much smaller. It retains the tumor targeting and uptake of the intact parental antibody, but because it's lower molecular weight and importantly does not have a full FC region, it targets and clears very quickly, and that makes it ideal for imaging. And over the years, we've been exploring this both for radionuclide imaging, such as PET imaging, and also for optical imaging by labeling with fluorescent dyes for intraoperative work. And we also looked at, can we use this to deliver therapeutic isotopes in order to actually treat cancers, not just find them? So this is a study we published a couple of years ago where we radio labeled that mini-body, PSCA specific mini-body with iodine 131, and looked, that's a hard beta emitter, so it's a therapeutic isotope. And we looked in a xenograft model in mice of 22 RV1s that have been transfected to express PSCA. And what you can see is as we increase the amount of radioactive anti-PSCA mini-body that we're administering to the mice, we see an increasing dose-dependent inhibition in the tumor growth and improve survival, as you can see here on the right. So that all looked very promising, but this particular molecule showed higher kidney uptake than we would have liked. And that would limit the utility of this prototype fragment, especially when using radiometals, where that radioactivity is cleared via the kidneys and then remains in the kidney and can deliver unwanted radiation dose. So now I'm going to turn to these middle-sized, intermediate-sized fragments, single chain FVFC antibodies. So you can see we have 2 antigen combining sites, but we've kept the full hinge at FC region. And so again, they retain the tumor targeting and uptake of the parental antibody, but they have tunable pharmacokinetics, which I'll go into in a little more detail, but also because they are so large, 110 kilodaltons, these definitely clear via the liver, which is much more radioresistant. So let's look a little more closely at our lead candidate and how we got there. Here is the single chain FVFC. And what's important is that the half-life of antibodies is controlled by this FC region. It binds to a receptor called the FcRn, which recycles and maintains antibodies in the circulation. And so what you can do is you can introduce mutations into that FcRn binding site. So these are the mutations here that knock out the ability for the antibody to be rescued by the FcRn. And the result is you have accelerated clearance. So let's look at our lead candidate. It is an anti-PSCA single chain FV. These are humanized variable regions. And then the FC region of IgG gamma 2, but now with 2 mutations that accelerate the clearance. So it's fully humanized. It has high affinity for PSCA, about 1 nanomolar affinity. The molecular weight ensures hepatic, not renal clearance, so that's better for overall normal tissue toxicity. And those dual mutations drive accelerated blood clearance. 12-hour half-life instead of, I think the wild type is more like an 80-hour half-life. So this format should be able to deliver rapid tumor targeting while sparing the kidneys and the bone marrow from unwanted radiotoxicity. And that should allow us to administer higher activities overall and deliver higher doses to tumors. So here's some preclinical work where we've evaluated the ability of this lead candidate to target to tumors in vivo. So one of the tools we use, of course, is non-invasive imaging, PET imaging, using the radioisotope zirconium-89. And so this experiment is actually a knock-in mouse model. So it's a transgenic mouse that has human PSCA in it, so that these tumors are not immunogenic. And we have a PSCA positive and a PSCA negative tumor in the mouse. And what you can see is that over time, the candidate antibody shows rapid targeting and uptake and accumulation in the positive tumor and less so in the negative tumor. And you can also see that the blood clearance is very quick. The body of the mouse is very clear by 24 hours, and it has indeed cleared to the liver as we expected. And this is compared to the wild type, which over time you can see you do get that good localization, but there's still a lot of blood pool activity in the 4-hour and 24-hour images, and this is going to give us undesirable bone marrow toxicity. So to summarize the in vivo characteristics of the lead, the A2 single chain FVFC exhibits high specific targeting and uptake to the tumors that express PSCA, faster blood clearance, and as planned shows liver clearance to reduce potential activity and toxicity to the bone marrow and the kidneys. So what's next? Well, let's move on towards therapy. So what we've been able to do more recently is that we switched from labeling with an imaging radioisotope to lutetium-177, which, as I mentioned earlier, is a hard beta emitter for targeted radionuclide therapy. And we've conducted full biodistribution studies where we looked at the uptake and accumulation of the radio-labeled antibody in tumors as well as normal tissues. And based on those, we've developed time activity curves and can calculate and estimate what would be the radiation absorbed dose in these normal tissues, such as liver, kidneys, and bone marrow, and also what could we deliver to tumor. And based on those results, we've actually now conducted a preliminary therapy study. This is in nude mice with 22RV1 PSCA tumor xenografts. They were given 1/2 millicurie of the lutetium DTPA labeled anti-PSCA single chain FVFC protein. And what you can see in this graph on the right is that we see a significant delay in tumor growth in this model. So we definitely can deliver activity and have a therapeutic effect. So in terms of the next steps, this is a great start. We have more work to do. We have shown that our lead candidate demonstrates the desired targeting, pharmacokinetics, and clearance properties ideal for targeted radionuclide delivery. And our preliminary study demonstrates tumor inhibition in a prostate cancer model. So what we're doing now is taking that agent now back into the syngeneic knock-in mouse model and looking not just subcutaneous, but also orthotopic models. But in parallel, we are working on how we can translate into the clinic. We're working on developing a stable cell line and a master cell bank so that it can be manufactured. I'll just mention, fortunately, at City of Hope, we actually have amazing, CGMP manufacturing facilities, so we actually can generate this protein in-house. And then our goal over the next couple of years is to conduct the IND enabling studies with the target of doing a first-in-human PET imaging study in patients with metastatic pancreatic cancer. We want to have a first look at what is the targeting capability and what are the pharmacokinetics and what does the dose estimation look like in patients before we switch then to a therapeutic radionuclide. And that, of course, is the principle of Theranostics. You can use the same agent and image first to evaluate target expression, accessibility, delivery of your therapeutic agent, and then just flip it to a therapeutic radioisotope and have a very specific, highly targeted, localized radiation therapy system. So this work is currently funded by a NCI R01 grant, but we, of course, would be looking for future funding to accelerate this work, in particular to get it into the therapeutic realm. So to summarize, we have a distinctive target. There's no currently marketed therapies targeting PSCA. We have a unique carrier, an engineered antibody optimized for pharmacokinetics and safety and efficacy. We have a couple of diseases where there still is a huge need, prostate cancer, where much of the existing focus on recent and new therapeutics is on PSMA, androgen receptor, small molecules, and immunotherapy. But there already is tremendous interest in targeted radiopharmaceuticals in prostate cancer, with Xofigo and Pluvicto being approved. And then pancreatic cancer, it goes without saying there is a huge unmet need for new therapies. So I think to have this kind of specific targeting agent for pancreatic cancer would be very important to pursue. So I'll stop and would welcome your questions.
Fantastic. Thank you for sharing. That's some great work there. Anna, you missed something in your intro. You have some experience doing this before. So I'm going to start off with a softball here for you and ask you to introduce some of the work that you've done in this space.
Yes. So,
yeah, go ahead.
Yeah, sure. So what I didn't say in the introduction is that a number of years ago, actually, Rob Ryder and Chris Behrenbruch and I co-founded a company called Imaginab. And the purpose of Imaginab was to use that concept of these engineered antibody fragments to develop targeted imaging agents and to develop and translate and commercialize them. And over the years, initially we looked at tumor targets, but more recently we did a pivot, and this will resonate with the earlier two speakers, because we realized one of the huge unmet needs in the cancer imaging field would be the ability to look at what's going on in immunotherapy. So we developed an imaging agent for CD8 T cells, which are the actual worker bees in most immunotherapies. The cytotoxic T cells are the ones that you need to actually eliminate your tumor cells. So we have a human CD8 specific mini body that is now in phase 2B. It's looking very good. We actually are presenting some results this week at the SITC conference, showing correlation between what you see in the PET signal, where we can image the CD8 T cells in the patient, versus what's in the paired biopsies. So our phase 2 studies look at a baseline pretreatment scan and biopsy, and then patients go on to standard of care checkpoint inhibitor therapy. And then a few weeks later, we look on treatment with this non-invasive CD8 PET imaging to see whether or not there's an increase, an expected favorable change in the T cells. So yeah, we've been through the process of how do you translate, and this is the worst kind of product you can think of, right? It's a biological and it's radioactive. So talk about a complicated product. It's a melting ice cube. Lots of fun. Anyway, questions?
I'll ask a question. Really nice work. I thought kind of taking us through and really interesting on PSCA. Just a question. Obviously, lutetium is well validated, in prostate, but I was curious. There's a lot of activity in kind of the actinium space, and so how do you think about your choice of isotope?
We're absolutely interested in actinium, and that is on the list of the current work as well. Right now, the lutetium is more available. There's more experience with it. But at City of Hope, where I co-chair the Theranostics clinical program, we actually have 2 in-house actinium-labeled antibody trials. And we have, I think, 5 industry trials in actinium. So we're definitely very interested. I think, so actinium is an alpha particle emitter. Beta particles have a longer path length, but they're lower energy, and they seem to have their effect through reactive oxygen species and things like that. Alphas have, an alpha particle is a helium nucleus, so you get a lot more bang for the buck, but it doesn't go as far. I think availability has been the challenge, but I think people are seeing, for example, with Xofigo, which is a radium-225, people have all of a sudden realized that these alpha particles can be the part of effective radiopharmaceuticals. And so in the field, there's been huge interest in actinium as an alternative. And I think we have to do the clinical studies. I think a lot of those studies are still phase 1, and there's still questions, about overall efficacy and toxicity. It's always a balance, but definitely that's on the list for the future, would be to look at, to see how well the beta performs, but then to also consider the alpha particle emitters such as actinium for this work.
Thank you.
Anna, can you speak a little bit to the overlap of PSMA versus PSCA expression?
You know, they both start out as really good targets with about 95% of prostate cancers expressing them. And I think one question is, once you treat with PSMA specific targeted agents, you wipe out those cells. There's still other cells that are left. You need alternative targets. And then I think another issue with PSMA, and especially with the actinium labeled small molecule, is the fact that PSMA, these small molecules do localize to salivary glands and lacrimal glands. So it does cause side effects that have caused patients to stop treatment. So one of the things about PSCA is it's got a complementary specificity, and it's not going to affect salivary glands, for example. So I think there's still plenty of room for cocktails or sequencing of some of these targeted these antibodies that target tumor targets, plus the additional benefit that we can start to look now at pancreatic cancer and bladder cancer as well with this agent. So that's another way that I think this is would be a valuable target to go after.
Any other questions? All right, I got one more then.
Okay.
So keep going. We got a little bit of time. You mentioned that PSCA is a glycoprotein. Any changes in the glycosylation state of either at different stages of progression, or does your antibody sort of recognize the glycosylated form, or is it sort of agnostic to that?
I think it recognizes a protein epitope, not the glycosylation.
Yeah.
So that really hasn't been an issue to worry about glycoforms on this protein.
Great. Okay.
Okay, thank you.
Thank you. All right. To close us out today, our final speaker is Denise Montel, the Dugan Professor of Molecular and Cellular and Developmental Biology at UC Santa Barbara. And Denise will be sharing her work today on engineering macrophages to fight difficult to treat cancers. So thank you, Denise. Denise, we could see your slides before.
Yeah,
and there you are.
As soon as I shared my screen, I lost the ability to unmute. So, okay. Okay. So Zoom disappeared. Anyway, well, great. It's wonderful to be here, and thank you for inviting me. I'm happy to tell you about some of our work. Now, I'm a basic cell biologist, and in my lab, we're interested in how cells behave normally when they're building, maintaining, and regenerating tissues, and then how they misbehave in diseases like cancer. And then our hope is to harness that knowledge for new treatments. So today I want to tell you about a new immunotherapy approach that has its origins in work that we have been doing for about 30 years and starting in fruit flies. So, but where we're going with this, I'm sure all of you here, I mean, we've already heard it several times today, are aware of CAR-T, but you may be less familiar with its sort of emerging younger cousin, CAR-M, or sometimes called CAR-P immunotherapy. And so the idea here is that you similarly have a chimeric antigen receptor that recognizes a target on a tumor, but now we're expressing that in a macrophage, hence the M, or phagocyte P. And the idea is that activating the receptor because of the domains that are used in the CAR-M therapy will trigger the phagocyte to engulf the target cell. Okay, and so, but not only that, what happens is that it will trigger an inflammatory signal. And this has beneficial effects because the tumor is actually being physically removed. But in addition, once those targets are digested, it's going to result in antigen presentation to the adaptive immune response, secretion of cytokines, and generally, hopefully, activation of a broader T cell response than you would get in CAR-T, because now all the epitopes in the tumor are available for presentation. And then, of course, there's also the ability for these macrophages to stimulate inflammation, and then eventually also to be involved in resolution of the inflammatory response. So I first heard about CAR-P or CAR-M from a faculty candidate who came through our department named Megan Morrissey, who was at the time a postdoc in Ron Bale's lab. And she showed us nice movies like this. These are macrophages in green and target B-cell lymphoma cells in magenta. And if you don't express the CAR in the macrophages, they sort of interact with the cells but don't do anything. But if you express the CAR, you can get these occasional whole cell engulfment and killing events, which was very exciting. And in fact, you may be aware that there's a first-in-human trial going on with CAR-M for breast cancer through the company Charisma out of the University of Pennsylvania. And it appears so far to be safe, but probably not as effective as we would like. And one of the challenges here is that the macrophages tend to nibble on the cells rather than totally engulf and kill them. And so what Megan Morrissey said at her job talk was that what they would really be interested in would be something that would cause these macrophages to be more aggressively engulf and kill the tumor cells. So now if we think about how would we possibly go about this, we can look at the signaling pathways that are activated in a macrophage when it engulfs a target. So here we have a target, we've got it bound to antibodies, and here we've got the FC receptor on the macrophage. And this is a simplified view. There's really about 150 proteins involved in the sort of engulfment signaling pathway. So it wouldn't be necessarily clear exactly where to start, but it turns out, I want to point out that one of the proteins here is this little one called RAC. And this is a protein that my lab has been working on since I started as a new assistant professor at Johns Hopkins Medical School in 1992. So in 1992, what is RAC? It's a protein. It's a very powerful little 21 kilodalton member of the RAS superfamily of GTPases. And this landmark paper by Anne Ridley and Alan Hall in an issue of Cell that came out the very month I started my job showed that if you take a serum starved fibroblast and you inject an active version of RAC into it, it really activates the cell. Not if you had a movie, it makes the cell ruffle around the edges, polymerize actin, and importantly, I will give you a little foreshadowing, triggers macropinocytosis, which is a type of engulfment. It's a fluid engulfment. So now I was starting my lab because I had developed an in vivo model to study cell motility. And as I mentioned, this was actually in fruit flies. And when that paper came out, we thought we didn't know much about cell migration, but we thought maybe RAC has something to do with cell migration. And so RAC is deeply conserved in evolution. Its original form probably arose in bacteria. So flies have RAC, humans have RAC. And what we did was we have this set of cells in the fruit fly ovary that are normally migratory. And when we inhibited RAC in these cells, they completely failed to migrate. But the important observation that remained a mystery for us for a very long time was that if we expressed the activating mutation, and I'm talking the same activating mutation that Anne Ridley and Alan Hall used in their experiment, it resulted in the entire tissue dying. Okay, well, now we're only expressing the protein in these little cells right here in 6 cells, and the whole tissue dies. And you don't have to be very familiar with fruit fly ovaries to tell that this is like dead. Okay, so the question was sort of why and how was active RAC like resulting in this tissue destruction? And I will make a 25-year story, cut it short. This was our cold case for many years. And what we figured out is that what active RAC does when it's expressed in a cell is cause it to eat other cells alive. So in this example, you're looking at a cell expressing active RAC. Normally without the active RAC, the cell is living in harmony with its neighbor, but here it's engulfing the cell next door to it. Now this is in a fruit fly ovary. But we made the leap and said, well, would active RAC potentially do the same thing in enhance the phagocytic capacity of a human macrophage? And so this experiment was done at a postdoc in my lab. He took a HL60 derived macrophage expressing an active form of RAC and co-cultured living human Jurkat T-cell leukemia cells. And basically they gobbled up the T-cell targets. And so this is a quantification of those data, the control level of engulfment, You can stimulate engulfment just even with wild type RAC, but you can stimulate it a lot more with an activating mutation. So active RAC is sufficient to cause cells to eat other cells alive. And our question was, could RAC potentially enhance, be the thing we're looking for to enhance the phagocytic capacity of the CAR-M or CAR-P macrophages? So the idea is just that we co-express in the macrophage the CAR and the RAC. And so it turned out that I collaborated with Megan Morrissey to do that because remember I told you that she told us about CAR-M on her job talk. Well, she was our top candidate. We made her an offer. She had other offers, but she fortunately joined our faculty. Unfortunately, it was March of 2020. So it took a little while before we could actually do the experiment, but I'll just remind you that if you have a macrophage without a CAR, it doesn't eat. And if you have a macrophage with a CAR, it engulfs some whole cells. But if we now add the macrophage, the CAR, and an activating mutation in RAC, then what we see is that the macrophages become hyperphagocytic and they engulf and destroy many tumor cells. So this was exciting, and we call this race CAR for RAC enhanced CAR therapy, and this is something that we have filed a patent on, and that it has been licensed out to by a tech company that actually launched a subsidiary company in order to pursue this as a CAR therapy. But I think there's more here. And I do want to point out that one of the things we're excited about is that I want to point out that that company is relatively small and its effort is pretty focused, and these are still early preclinical days for us, but I think the potential is very broad, and I'd be super interested to talk to Anna about using her antibody and engineering a CAR-M with her antibody. So against pancreatic cancer or something like that would be really fun. We should collaborate on that. That would be cool. But I also want to point out that the effect of RAC on the macrophages is pretty cool because it only enhances the phagocytosis of the specific target when there's a receptor ligand interaction present. So you get to tell it what to eat. It's not going to run around and eat just anything. So data for that would be here. I showed you the movie. If you have no CAR and there's a wild type RAC gene, you get no eating. If you have RAC, the activating mutation in RAC, but no CAR, there's also no eating. And if you have the CAR without the, with wild type RAC, you have a little bit of eating. And if you have the CAR with the activating mutation in RAC, you have greatly enhanced eating. But what I want to just finish up quickly with is another idea that we're really interested in and looking to partner with folks on. And that is, as we all know, there are challenges, although immunotherapies are very exciting and there's hundreds of companies around them, there are challenges to them. And the challenges to all the immunotherapies, CAR-T and CAR-P, are that they're complicated and expensive. And this limits really the number of patients that you can help. So we envision that we could solve all of those problems and create really an off-the-shelf race CAR simply by delivering instead of taking the cells out of the body, engineering them in the lab, expanding them and putting them back into the patient, we are envisioning that we could encode the CAR and the RAC on mRNA molecules that would be embedded in lipid nanoparticles, similar in concept to a COVID vaccine. And the great value of this would be that you could actually engineer these for all kinds of different diseases, and they wouldn't have to be made individually for each patient, but rather they'd be an off-the-shelf therapy that you could deliver. And there's been some, interest in this kind of approach for CAR-T. And but I think that I think it's going to be massively better in CAR, and because macrophages are going to be easier to deliver these things to specifically, because macrophages are specialists at eating and expressing what they eat. And so, and there are, you know, we have ideas about how we could engineer these nanoparticles to be relatively specific for macrophages and not be engulfed or and expressed by any other cell type in the body. So we're definitely looking for partners to develop an off-the-shelf sort of race car. And I will stop there. This is pretty much the view out the window of our lab, and acknowledge that there have been really a couple of people, this postdoc Abhinav Mishra and a graduate student Melanie Rodriguez, who have been the real leads on this project. And I will take your questions.
Excellent. Denise, thank you so much for sharing that and for making me incredibly jealous of the view outside your window. Really great work. I think, you know, in our preview call for this, right, the question that I had was, can you overdo it, right? Can you have too much RAC activation and, you know, kind of get those macrophages overstimulated and eating the wrong things? I think the data that you showed there is quite exciting that doesn't seem to be the case. Thinking about indication selection, right? There's been challenges with cell therapies in solid tumors. Macrophages obviously have some advantages there. Kind of where do you think this is best suited to go, and where would you put it first into the clinic?
Yeah, I mean, you know, we have a lot of preclinical work to do, but that's one of the reasons, like, I would really like to see a bigger effort on it, because I think we have since macrophages are specialists at wriggling into every nook and cranny and every organ and tissue in your body, I sort of feel like they're going to do a better job of getting into tumors. And that's been a major obstacle for the CAR T therapies, obviously. We also see, we've done RNA-seq on our RAC enhanced cells, and we see signs of things being upregulated that will make them even better at getting into solid tumors. So we're very excited to try it against solid tumors. And it's a really good question. Like, what do you go for first? Do you go for the thing that might be the lowest hanging fruit, like the thing that's most likely to work because that's most likely to give you a success that will fund a further effort? Or do you go for the thing that's where the need is highest and you know, take your chances with that? And I think in the preclinical model, there's absolutely no reason we can't try lots of things. We can try lots of different CARs and our RAC enhancement of lots of different CARs against lots of different tumors. And I sort of had it in the slide, but I think if we really could get this lipid nanoparticle thing going, it wouldn't only be cancer anymore. And like, cancer is huge and we want to do, you know, we definitely want to do that. But if you made it affordable, well, first of all, with an American Cancer Society goal of making things more equitable, if you made it more affordable, it certainly would be more equitable, right? So many, many more people could get treated with these cutting edge therapies if we take that approach. So I think there should be a huge investment in it just for that reason. But in addition, you could target, like, macrophages will eat viral infected cells or antibiotic resistant bacteria, or, you know, all kinds of things, you know, maybe tau tangles, for all I know, you know, they'll eat anything you tell them to eat. So I think that the potential is really very broad, and it's a matter of just, you know, resources to see just how broad.
Mm-hmm. Any questions from the group? All right, Denise, I got maybe another one then for you. In addition to, you know, the story of RAC activation here is really interesting, and you didn't get the chance to talk about some of the history of that. Maybe since you have a minute, I know the arc of your research is incredibly interesting here. Maybe share the short version to give a little bit of that context.
Yeah, no, I mean, I hinted at it, like, you know, the first experiment we did when I was an assistant professor was test the role of RAC in cell migration. And, you know, that was cool. And then, many years later, we made a photo activatable, like an optogenetic form of RAC, and we can use it. It's like a little video game. You can steer cells around tissues with this opto form of RAC. And all of this led to this dominant model in the field where local activation of RAC in a cell causes local actin polymerization and really sets the leading edge for a migrating cell. And that's true for cells, immune cells, cancer cells, fruit fly cells, like cells, dictyostelium, amoeba, like, any kind of stuff. It's a very fundamental cell behavior. But like I said, it was just a mystery for 25 years as to why active RAC seemed to just like destroy the whole tissue. And like, how do 6 cells destroy this whole tissue? But I want to say that from a cancer treatment angle, that's encouraging. Like, you can put this active RAC in a few cells and you can gobble up a lot of and kill like a big thing, like maybe a tumor. And so it just took us a long time we needed pieces of information that we didn't have to piece together the idea that what might be going on is that the cells might be actually eating to destroy the neighboring tissue. And that really came from other people's work showing that that's a natural thing that happens. These cells in the ovary sometimes have to eat their neighbors, either part of normal development or in response to stress. And that put the idea in our head that might be what was going on. But of course, that would have just been a little fruit fly story, except that about the same time we figured that out, these human patients came along. There's a paper published about human patients carrying activating mutations in RAC2 and the sort of disease that they have. And there was a mystery there. Nobody understood why some of their cells were disappearing. And we figured out, just based on our fruit fly work, we were able to figure out that the basis of the disease that's caused by activating mutations is that the patient's macrophages are consuming their B and T cells. And then the CAR-M sites. That still would have been a pretty limited interest, but then we realized we could harness it for CAR-M. And so, yeah, so it's a 30-year story.
Fantastic.
Fruit fly ovaries to cancer treatment.
Fruit fly ovaries, reverse translation from a patient population. So really incredible work there.
Yeah. Denise, fantastic story how you got here, but also I love your cars and we definitely should talk.
Yeah.
Have these gone in vivo and 10 in vivo models?
So the Charisma has worked on it, has an in vivo model without the RAC, right, just with the CAR, and it looked promising, and they published that. And then they have this first in human trial as a safety trial right now. We have not as yet gone into mice, and I'm mostly a fruit fly lab. We do some mouse work, but I'm interested in collaborators to work on that too.
Yeah, we should talk.
Yeah, totally. Would love to.
And Denise, you alluded to the company that is progressing some of this work, maybe just for clarification and to reiterate sort of, what the obligations to the licensee are and sort of what is still.
Yeah, I mean, as far as I understand it, because again, I'm not really an expert in IP and stuff like that, but as far as I understand it, so they've licensed this, race car technology. They don't even actually call it that, but I like it. So, and so I think they are not pursuing, we definitely in collaboration with them are pursuing some of the CAR, straight up CAR P, CAR M approach with RAC, but they are not pursuing the RNA lipid nanoparticle part of this. And so I think it would probably constitute some sort of sub-license or something, whatever you call that. But I think it would be available because they're a cellular immunotherapy company. They're not going to be pursuing that angle. And I do think from the democratization of this whole thing, it ought to be pursued.
Yeah. And since it's public information, I don't think you've mentioned it, but it's Inceptor Bio, right? That has licensed this, and they're also going and probably looking for funding in this environment. I would guess.
Yes. They got some funding, and but I'm sure they need more.
Well, or they will at some point. That's inevitable.
Mm-hmm. Yes.
Great. Well, Well, we have a couple more minutes left, and I definitely wanted to make sure we kept this to time. So maybe I'll just take this opportunity to kind of reiterate, because there's definitely some asks to the group. I think this is always a great opportunity to remind everyone. So Brian Eliceiri had the opportunity to share with us his work on agonism of the alpha-7 receptor and how that activates antigen presentation and synergizes with PD-L1 treatment. Definitely some opportunity there to support further understanding of the already available molecules and maybe move those forward, potentially to create novel chemical entities that also are involved in this agonism. Crystal Marconett shared with us the great work on her long non-coding RNA that correlates with tumor mutational burden that can be used as a patient selection biomarker. Great opportunity there to collaborate and look for a retrospective analysis of PD-1 treatment and to see if that biomarker does validate in that setting. And Anna Wu shared with us the use of a PSCA, pharmacokinetic optimized antibody fragment, for use in both a theranostic approach there, and looks like that is ready for IND enabling studies and really is off to the races. And then we just finished up with Denise Montel describing her race car technology, one licensee that's got some of that work ongoing, but some opportunities to further expand that work as well. So happy to wrap it up there. If there are any questions to any of the other presenters at this point, I know Brian, we cut you off a little bit short in the middle of the Q&A, but we got ourselves back on time. So take the last 5 minutes here if there's any other questions or follow-up. Steve Gilson, any other housekeeping items or expectations coming out of this?
Just that I'll send an email either later today or tomorrow morning with a link to the video of this presentation. And as always, if anyone wants to connect with each other, please let me know. I can make those introductions. I do not send out the whole emails. All the presenters' emails will be sent out because that's the point. But I will not send all the attendees' emails out. If you want to get connected with people, please let me know, and I will make those introductions. But otherwise, and thank you again to Alexandria Venture Investments, who is the primary underwriter of this program, as well as to, of course, the great collaboration with Bioscience LA and American Cancer Society and Bright Edge Fund. It's been really amazing to work with you guys on this one. So excited to have you guys involved in the Southern California ecosystem and leaning in and helping really help commercialize the great things that are working in this region. So thank you very much, really. And thanks to all the presenters, of course, great work, and for everyone attending. Thank you for being here.
All right. Thank you, everyone. Great presentations.
Thank you.
Great discussion.
Bye. See you.